All right, so today we are talking about a little bit with the scientific method because we're going to have a couple labs coming up where this is going to be one of the fundamental parts of science. So I know you guys generally kind of talk about this every year, but I just want to re-hit a couple things. The main focus for us is going to be on variables, okay? So hopefully none of this sounds like completely brand new, but you guys know when we're looking at a scientific experiment, somebody at some point went, right? You guys sometimes have that moment like, right? Even as simple as how many people walked outside this morning and was like, ooh, it's kind of cold. Yeah. And so when I walked out, I'm like, ooh, it's kind of cold. So I made an observation, I turned right back around, and I went and got a jacket before I left. Especially if you're riding the bus, you probably like, I kind of want a jacket or some kind of zip, okay? So anytime that you guys are using your senses, you're observing, and then you're making decisions based upon that, that in itself is the scientific method. It's just for us, because science is so rooted in things like engineering, design, medicine, there has to be kind of like a strict procedure so that I'm not just creating a new medication and being like, it's going to fix cancer, and it has zero effect on it, right? So our whole process kind of goes with, I want to solve something. I observe something, or there's a question I want to try to answer, so I come up with a hypothesis. Do you remember what a hypothesis is? Is a educated guess, right? And it's just a guess. It's okay. Is it all right if at the end your guess was wrong? Yeah, totally. It's perfectly fine. Okay? So then we're going to experiment to test our hypothesis and then analyze our results and then come up with a conclusion. It's okay if my conclusion is that my hypothesis was wrong, right? That's all science is about, is going through, observing something, having that moment, and saying, how can I fix that? How can I make that better? That's what makes us as humans so unique compared to the rest of the animal kingdom. So we're constantly looking at innovating, designing, improving, okay? So we're constantly taking in with our senses our natural world and trying to be like, well, how can I make this better, right? So that's kind of the process that we are going through. With the hypothesis, I know you guys all know it, you can regurgitate it. It's an educated guess, right? Usually we write it as if-then statements. The big thing with a hypothesis, though, is it has to be tested. I have to be able to test it, right? If I make a hypothesis about the origin of the universe, can I test that? No, because I can't recreate the start of our universe. There's like no way for me to be able to do that. Now, could I test to see how my bonsai tree will survive this year? We're really going to shoot that my tree doesn't die. That's the goal. I don't have a really good green thumb, but we're going to see. Maybe it'll live. Maybe it'll live. So usually the if-then statements is like, if I water my plant enough, then it will grow and continue to live, right? So I usually have an if, independent variable, then dependent variable. So this is going to help me predict an outcome going forward. Now, in an experiment, I know plants. What are some things that affect a plant's growth? Sunshine, water, oxygen, carbon dioxide, soil, size of the plant, humidity. So there's a lot of things that are really going to impact whether or not that bonsai tree makes it to December, right? There's a lot of different things. Even like room temperature. Room temperature could affect it. So can I test all of those things at the same time? No, I can't, right? I can't test all of those. So in order for it to be a valid experiment, I'm only going to change one variable. Just one per experiment. Could I test water and then do another experiment later and change the amount of sunlight? Yeah, but at that point, the water has to be the same for all of them. So there is a way that I could figure out what is the best combination of all of them, but I would have to do those each in a different separate experiment. Because I want to be able to say specifically the water caused this. The sunlight caused this, right? I want there to be direct correlations, which hopefully last year when you guys took bio, you know how, especially around the air test time, you looked at so many graphs. You looked at population trends and you saw how those changed. So when we're looking at experiments, we're gonna eventually try to say this led to this. There's a direct correlation between the two, okay? So the one thing that I changed the independent variable. What Roman numeral is this? 1. Ooh, 3. So this is Roman numeral 1, right? Independent variable, I changed it. There is one thing that got changed, right? So that's how I keep track of it. So independent, it's what I change, I being the scientist, and it's only one thing. I can only change one thing at a time. So one thing is the dependent, and then what effect that has is going to be the dependent. So dependent, I'm going to measure this or observe it. So dependent is what I measure. So if I change the amount of water, what could I measure? I can measure whether it's alive or dead. I could measure its height. I can measure the number of leaves. I can measure number of flowers, things like that. So independent is what I change. Dependent is what I'm measuring or hoping to see that impact on. Okay? So again here, this is the one we've been talking about with the plant. If I change the amount of water, I hope that it's going to be bigger. It's going to have more leaves. It's not going to be dead, right? So I know independent, I can only change one. Can I measure multiple things with the same experiment? Yeah, totally, right? So I can measure a lot of things. Doesn't necessarily just have to be one, okay? All right, there is one other variable that you guys will hear, and that is the control variable. The control is what you are going to keep the same. If I have 5 plants and I'm going to change the water for each of them, can I have one of them be a sunflower, one be a redwood tree, and one be like a hosta? No, that's something else that I just changed. So I got to keep the type of plant the same. They're all going to be sunflowers. Should they all be in the same pot size? Should they all be in the same location? Should they all have the same humidity? Same amount of sunlight? Yeah. So those are what I would call the control variables. So control variables, they are factors that we know can impact the outcome, but I want to make sure that they're the same across the board so that they are not influencing my results. So again, the control is not what's being tested. I'm just keeping that the same across the board for all of those points, okay? Because the controls can affect my results, but if I control them, then they're not, okay? All right, so how many of you guys are starting to drive home with a friend or a sibling? Yeah. Have you guys ever been out into the student parking lot? The goal is to get out of here as fast as humanly possible, right? And so sometimes they go out by Memorial. Sometimes they cut across the front of the school to hop out. And then sometimes they go behind the school, right? You guys ever notice that? It's like a mad dash. So if we change the route, we want to be able to measure then the amount of time it takes. Because I want to get out the fastest, right? That's my end all goal. I'm a junior, senior. Dude, I'm out of here. Piecing out 232 as fast as I possibly can. So if I'm going to change the route and measure how long it takes to get there, what do you think are some controls I might need to keep?
Having the same car.
Yeah, I should probably have the same car, right? What else? Same time, good. I should leave at 2:32 exactly, maybe 2:33, because it takes me a little bit of time to get to my car. So I leave at 2:33 every single day, okay? What else? Well, no, I'm changing the route, right? That's what I'm going to test.
Same driver.
Same driver. How about number of passengers? Should I keep that the same? Yeah, okay. What about driving different speeds? Can I drive 20 miles per hour that way and 50 behind the school? No, I should maybe keep that the same too. Because obviously, if I'm driving 50 and 20, the 50 is going to get there faster, right? Excuse me. So those would all be things that then are the same. I'm keeping it the same, hands down, so that I know that by the time I'm done, whichever route that ends up going the fastest is the best route, okay? So that will help me get out of here a little bit quicker, okay? So again, changing that route, that's my independent, that's what I changed. It's the one thing that is unique, that is different. And then what I measure is the dependent, which on this one I'm looking at time. And then my control, again, just the route, or keeping things like the car, the number of passengers, how fast I'm driving, what time I leave, okay? So ultimately, I want to have a valid experiment. So in order for it to be a valid experiment and for me to make a claim at the end and say, hey, this does or does not support my hypothesis, I got to keep this very structured, okay? Now, how many of you guys are thinking about medicine? Is anybody thinking about going into something medical related? All right, so yes, some hands. All right, so in medicine, let's say we're trying to find a new cancer drug, all right? We usually have an experimental group, and then we also have a control group. Which one gets the new chemo drug? Experimental group, right? That's the new drug that I'm testing to see if it has an effect on cancer. What does my control group get? Nothing, right? So when we're looking at those new medical studies or a new research group, right, we're looking at I might get a new miracle drug or I might get nothing, right? So like if it's a pill, they probably get a sugar pill. If it's an IV, they might have just saline. And then that's it, okay? So they're getting that placebo. So that I can say, hey, this drug actually reduced the tumor size comparative to the control group, okay? So sometimes, especially in medical experiments, we're looking at having an actual group of people that we would classify as the experimental group, and then a group of people that are the control group. Now, obviously, people, we're not all identical, right? So there are going to be other factors there. But if the entire experimental group saw a 50% reduction in tumor size, and the control group didn't see a reduction, or maybe they saw an increase in the size, then we would be able to say, okay, this was effective or it was not effective, okay? All right, so what do you guys think about this one? So take a look at this example. So Sarah's learning how to drive, and she really wants to determine how fast she can go in 10 seconds. She thinks the car will go faster if she has bigger tires on it, okay? So Monday, she tries 3 different tire sizes. On Tuesday, she tries 3 different brands. And then on Wednesday, she repeats Tuesdays, but it's raining that day. Would you count this as a valid experiment or not valid? Not valid, right? Because she's changing what? Yeah, she's changing the independent variables multiple times, right? So she's changing the tire size, which is what she originally thought her hypothesis was. She should have just repeated Monday like 3 times. See if there was a difference between them, right? Tuesday with a different brand. So we changed now tire size and brand, and then also weather on Wednesday, right? If it's raining, are you going to be able to go really fast? No, not at all. Not at all. So by the time we've done this experiment, and hopefully, again, we structured it so that it's a nice, valid experiment. At the end, I can make some good conclusions. But in order to do that, I got to collect my data. And so that data could be either quantitative or qualitative. So quantity, if you think of quantity, that's a number, right? I always remember it by the N being in the middle. That's a number. Whereas qualitative, that describes the quality. So I've said, what's the quality of your shoes right now? Are they brand new? Are they old? Are they muddy? Do they have holes in them? Okay? So quantitative, quantity, it's going to be numbers. So number of flowers, height of the tree, duration, time, age, distance. Those are all numbers that I can collect. But sometimes it's going to be qualitative. Okay? Who's tired today? Yeah, right? Okay, so if I said qualitative, how do you quantify tired? can you? No, it's a feeling, right? So sometimes some of our observations, or the data we're going to collect, are going to be in the form of words, right? How tired do you feel? Do you feel tired, or are you awake, right? Are you really tired, or are you just like kind of tired, right? Those would be all things that I could ask you and then figure out, well, how many students actually are tired versus not. Qualitative, we could use happiness rating, we could use pain scale, we could say eye color. When I say tired though, like my tired is probably not the same as your tired. Or my tired might be different than somebody else, right? So qualitative, we gotta remember, not necessarily gonna give us numbers, but more so words, descriptions, observations, things like that. So once I get all that data, you guys know we did this last year with bio as well, but we're gonna generate some graphs and we're gonna look for overall trends. Sometimes there is no relationship between them, and that's okay. Remember, it's okay for an experiment to fail, to get to the end of the lab and be like, it didn't work. It happens. That's perfectly fine. But that means we've also learned something, right? So just because you fail at something doesn't necessarily mean you're not learning something about it, right? You have Thomas Edison and how many light bulbs he went through. It doesn't matter how many times that he failed. It mattered that he actually created a light bulb that worked, right? So when we're looking at data and we sometimes get some results and we're like, there's no relationship, there's no trend. This chemotherapy drug did not work. There was no effect on the overall size. It just takes us back to the drawing board. It allows for us to modify our hypothesis and create another experiment. And that's what it comes down to, is making those final conclusions and being like, hey, it did work or it didn't work. Either way, we learned something from it, okay? So usually we do modify and retest. If we were successful though, we're gonna share that with our peers. And so then our peers are gonna go ahead and test it as well. Did they get the same results as us, okay? If my hypothesis is well supported by all my peers, my hypothesis turns into a theory, right? Does anybody know after a theory what it turns into? Starts with an L. A law. So think like conservation of mass. That's the law of conservation of mass. Can I create or destroy things? No, mass is always conserved. It has to come from somewhere, right? Now, there are some things that I would think are theories, or I think would be laws, but aren't quite necessarily there because I can't prove it 100%, right? Think about the universe. What's the theory that states how the universe began?
Big Bang.
The Big Bang theory. It's a theory. I can't prove that 100% without a doubt. Nobody was there. Nobody had a videotape. Like, we can't. We have an idea, right? The universe is expanding. We know that it is cooling. So if it's moving away from each other, then at one point back in time, it was closer, right? If it's cooling, it must have been at one point in time hotter, right? So there's some conclusions I can draw from it, but still just a theory. Think gravity. Is that a theory or a law? It's just a theory. Can you guarantee that there's no other things in action that are making this fall? I can see the effect of gravity. Can I touch it? No. So do you see how there's limits sometimes to science? So we might have something like gravity, which you would think like, well, duh, something's going to fall to the earth because gravity's going to pull it down, right? But sometimes it's still just a theory, and sometimes in science, that's where it ends up hanging out forever and ever and ever. And that's okay. All right, so just to reiterate, again, we're going to have that question or that problem or that observation that's going to make us go, huh, or really wonder about. And that's going to lead to our innovation, right? So we're going to form our hypothesis, we're going to test it out, gather some evidence, and then go ahead and make that determination. Does this actually match? Is there a correlation or is there not?